Sruthi Narayanan. She's a graduate student with Vara Prasad at Kansas State. She'll be talking about comparative responses of wheat to high daytime and or nighttime temperature stress during flowering.
Good morning, everyone. Tonight, 1 billion people across the globe will be going to bed with a hungry stomach, and among them, 200 million will be children. These people need the justice to eat. To feed the 9 billion people in the world by 2050, wheat production needs to be increased by 60%. Several environmental factors limit the yield and productivity in wheat, and among them, high temperature stress is a major factor that contributes to decreased wheat yield and productivity. This high temperature stress is brought about by differential changes in daytime maximum and nighttime minimum temperatures. These graphs show the trends in increases in daytime maximum temperatures and nighttime minimum temperatures over years. The upper graph shows the increases in daytime maximum temperatures, and the lower graph shows the increases in nighttime minimum temperatures. From the upper graph, we can see that daytime maximum temperature increased by 0.88 degrees Celsius over the 100 years. And from this graph, we can see that nighttime minimum temperatures increased by 1.86 degrees Celsius over the 100 years. Climate models predict similar trends for the future also, which means that the increments in nighttime minimum temperatures will be greater than the increments in daytime maximum temperatures. Plants may respond differentially to increases in daytime temperatures and to increases in nighttime temperatures. Therefore, understanding the responses of wheat plants to high daytime or nighttime temperatures is critical to anticipate the impacts of climate change on wheat production and also to develop appropriate management practices. Therefore, the objectives of our study were to quantify and compare the effects of high daytime and or nighttime temperatures during anthesis, which means flowering, on biochemical, physiological, and yield traits of wheat genotypes. The experiments were conducted under controlled environments in growth chambers. We used 2 winter wheat genotypes, a heat-tolerant genotype Ventnor and a heat-susceptible genotype Cal92. There were 4 temperature regimes, which were optimum or OT, high nighttime or HN, high daytime or HD, and high day and nighttime or HDN. We had 4 different growth chambers, and each of these growth chambers received one of these 4 temperature regimes. High temperature stress was imposed at the onset of anthesis, and stress was continued for 7 days. Let's move on to data collection. High temperature causes accumulation of highly active molecules that contain oxygen. These molecules are collectively known as reactive oxygen species or ROS. Accumulation of ROS causes several damages called oxidative damages, and that includes peroxidation of membrane lipids, which leads to membrane damage. Therefore, we quantified ROS content and plasma membrane damage in leaves using molecular probes. Under non-stressed conditions, plants have a defense mechanism composed of antioxidants that scavenge reactive oxygen species and protect the cells from oxidative damages. In the time of stress, this antioxidant capacity decreases, which leads to accumulation of ROS that results in oxidative damages. Therefore, we quantified total antioxidant capacity in leaves using antioxidant capacity assay kit, and we used a microplate reader to read the absorbance values of leaf extracts. The light energy absorbed by a chlorophyll molecule can be used for driving photosynthesis, or it can be re-emitted back as light energy. This latter process is known as chlorophyll fluorescence. These 2 processes happen in competition, so any increase in one process will lead to a decrease in the other one. Therefore, measuring chlorophyll fluorescence will give us an indirect estimation of efficiency of photosynthesis. we measured chlorophyll fluorescence using fluorometer, and this instrument gives us different fluorescence parameters such as Fm, which is maximum fluorescence, or Fv, which is variable fluorescence, which is calculated as the difference between maximum fluorescence and minimum fluorescence. And based on theory, Fe by Fm indicates maximum quantum yield of photosystem 2 or photochemical efficiency of photosystem 2. And we measured leaf level photosynthesis using Li 6400 photosynthesis system. And all these biochemical and physiological traits were measured on day 1, 4 and 7 during the stress period and 2 days after the stress was relieved. And these yield traits were measured at maturity. The experimental design was randomized complete block with a split plot treatment structure. The main plot factor was temperature and it had 2 replications because we repeated the experiment 2 times, and these 2 experiments acted as 2 replications for the main plot factor. The split plot factor was genotype and it had 5 replications because we had 5 plants per genotype in each growth chambers, and these 5 plants acted as 5 replications for genotype. And data were analyzed using prog mixed in SAS 9.2. Now let's move on to our results. Firstly, total antioxidant capacity under high temperature stress. Here the x-axis is temperature regimes, which were optimum temperature, high nighttime temperature, high daytime temperature, and high day and nighttime temperature. The y-axis is total antioxidant capacity expressed in millimolar uric acid equivalent. And here we can see that total antioxidant capacity significantly decreased under all the 3 high temperature regimes. And in the second graph, we can see that reactive oxygen species content significantly increased by all the 3 high temperature regimes. Similarly, plasma membrane damage also significantly increased under all the 3 high temperature regimes. Another interesting point here is that the effects of high daytime and high nighttime temperatures were similar on all these 3 traits. Now let's see 2 traits related to photosynthesis. First one is maximum quantum yield of photosystem 2. And here we can see that maximum quantum yield of photosystem 2 significantly decreased by all the 3 high temperature regimes. And now let's see how this damage got reflected in the rate of photosynthesis. Photosynthesis significantly decreased by the high temperature regimes. And now let's see the yield traits. The traits listed here are seed set, grain number per spike, individual grain weight, and grain yield per spike. And here we can see that seed set, grain number, and grain yield significantly decreased by all the 3 high temperature regimes. The decreased seed set percentage under high temperature regimes may be due to the poor performance of pollen and/or ovule. The previous reports from our own lab suggested that the major reasons behind decreased seed set under high temperature stress are the decreased pollen germination and decreased pollen viability. Decreased seed set is the reason behind decreased grain number per spike under high temperature stress. In this graph, we can see that individual grain weight was not affected by any of these high temperature regimes. This is because individual grain weight is the product of grain filling duration and grain filling rate. Stress was not imposed during the time of grain filling in this study. Therefore, grain filling duration and grain filling rate were not affected by high temperature stress. Since these 2 processes, grain filling duration and rate, were not affected by high temperature stress, individual grain weight was also not affected by high temperature stress in this study. And grain yield per spike is the product of grain number per spike and individual grain weight. Since individual grain weight was not affected by high temperature stress, decreased grain yield was the result of decreased grain number per spike in this study. Another interesting point is that both high daytime temperature and high nighttime temperature had similar impacts on seed set, grain number, and grain yield. But the combination of high day and nighttime temperature had increased or greater impacts on seed set, grain number, and grain yield compared to the high daytime temperature or high nighttime temperature alone. Based on the results from our study and the existing reports in literature, we propose possible mechanisms for yield loss under high temperature stress. High temperature, which can be high nighttime, high daytime, or high day and nighttime, decreases antioxidant capacity in plants. Thus, plants loses the ability to scavenge reactive oxygen species, and this leads to accumulation of ROS. And that causes several oxidative damages, and among them, one of the most important oxidative damages is the damage happening to membranes. And this can be damage to any type of membranes, which includes plasma membrane and thylakoid membranes. And all these changes leads to decreased photosynthetic rate. High temperature also damages the reproductive tissues of the plant. Poor pollen or ovule performance leads to decreased seed set, and that results in decreased grain number. And collectively, all these damages result in decreased grain yield. Conclusions: high nighttime temperature, high daytime temperature, and high day and nighttime temperatures increased reactive oxygen species content and membrane damage, and they decreased antioxidant capacity and leaf level photosynthetic rate. High nighttime, high daytime, and high day and nighttime temperatures decreased grain yield per spike through decreased grain number and seed set. Impacts of high nighttime temperature and high daytime temperatures were similar on most traits, but compared to high daytime temperature or high nighttime temperature alone, the combination of high day and nighttime temperatures had greater impacts on seed set, grain number, and grain yield. And the results from this research suggested that high nighttime temperature and high daytime temperature cause damage of a similar magnitude to winter wheat. Our future research will be focusing on evaluation of winter wheat germplasm for high temperature tolerance and also on breeding for high temperature tolerance using the identified genotypes from this study. And with that, I would like to thank my professors, our lab team, and our funding agencies. Thank you.
All right, we have time for a few questions.
Yes, ma'am.
Okay. So is your conclusion that high night and high day temperature have similar effects?
Yeah.
Because it seems like you use different level of temperature. Your night temperature was 24 degrees Celsius.
Yeah.
And then the daytime temperature 35.
Yeah.
If you use same temperature level, would you see the same effects? If you compare, if the night temperature is more detrimental or a high night temperature is more detrimental or high daytime temperature more detrimental, would you use the same level of temperature? Would you see different results if you use same level of temperature?
Okay, so the question is that in my case, the high nighttime temperature was 24 and high daytime temperature was 35. If I use similar high temperature for daytime and nighttime, what will be the responses? Okay, so my guess is that the damages due to high nighttime temperatures may be more pronounced than high daytime in that case. But I did not use that high temperature for nighttime because I thought in nature, usually what happens is that there will be some kind of difference between daytime maximum and nighttime minimum temperatures. So that's why I did not make it equal, high daytime maximum and nighttime minimum.
So that is true. The night temperature is lower than daytime temperature.
Mm-hmm.
That's if you don't compare it to I think we comparison.
Mm.
Then it is better to set at the same level of temperature, and then you can see if it's night or day, because the night and day temperature affects the respiration differently from other present.
Okay. Okay. Thank you. Yes.
Last question.
Yeah.
Question about the light intensity in the chamber.
Hmm.
How does it compare to the field levels, and if you think that having different intensities might change your results?
Okay, so the question is that how was the light intensity in my grow chambers and how did they compare with the field level light intensity? So the light level in my grow chambers was 900 micromole per meter square per second at 10 centimeter distance from the tube lights, and it was around 600, 700, or 700, 800 above the canopy. So I understand the light levels are a little bit low compared to field level, because field level maybe it's around 1200 micromoles per meter square per second. But I think in my cases, when I observed photosynthesis and those kind of physiological traits, optimum temperature plants had a little bit lower photosynthesis compared to what I can expect for the plants in field. I understand that this may be due to one reason, may be the slightly lower level of lights, but I think the levels I used was still okay to do the controlled environment studies. It was not that low, but I understand it compared to the field level, it was lower. But I got significant differences among the different temperature regimes, like optimum and the high temperature stressed one, because so I think the light level did not confound the results.
Thank you.
Thank you.